Hi, everyone, and welcome to the Trial Site News Podcast. I'm Dr. Erin, your host, and today we have Dr. Rupert Holms joining us from London, who is going to chat about Ezrin peptides and how they can be used to treat different viruses. And we're going to focus on long COVID for this podcast, but he'll offer some historical perspective because he's been working on these, created them for a long, long time. But thank you so much for joining us, Dr. Holmes. It's great to have you on Trial Site. And
well, Erin, you're very welcome. Thank you.
Yeah, this is a very interesting topic and research. Do you mind just giving us a brief introduction about who you are?
Certainly, I'd love to do that. Okay, I have a rather unusual sort of academic career history. And so I basically got a PhD from Edinburgh University in molecular biology a rather long time ago. I then actually went out of academic work and into finance. So I actually worked for a private Swiss bank, but I was managing life science funds. And in fact, then this is going way back. So we're talking about 94 when the HIV epidemic emerged. And so I actually started actively funding early stage life science research in HIV and HIV vaccines in the mid 80s. And from that program, we were originally looking for a sort of non-conventional vaccine programs. And we were working on two areas. One was looking at anti-idiotypic vaccines, that is, where an antibody recognizes a site that is being used by the virus, therefore the antibody is mimicking something on that virus. That was one strategy. And the other strategy we went after was looking at conserved sites, in this case HIV, which were mutation stable. And they're very important things on viruses because they have a functional significance. Particularly HIV mutates very quickly, and so anything that's stable is very likely to be functionally important to that virus. And even though it seems to be a complete tangential talk relative to long COVID, this is very important because this is the beginning of the therapeutic strategy. Okay, and the basic discovery was that the C terminus of GP120, the coat protein of HIV, mimics a human protein called Ezrin. And that Ezrin protein, in fact, when we first picked up the computer match for this protein and the structure in HIV, no one really knew what it did. You fast forward now 30 years, and we now understand these are essential organizing proteins that bring receptors on the cell surface together with cell signaling networks. So they are the center of a protein complex. And best way to imagine is like a piece of Lego, like a child's toy that you can plug different things into. And that's basically what Ezrin is. The issue about why it's sort of important, its activity is important, is because it is modulating different pathways, signaling pathways and different receptor usage. And so depending on the three-dimensional shape of those receptor structures, those complex protein complexes, you can determine different functions. Now, why was HIV coding for something like that? Well, it's fairly easy to understand, or at least hypothesize, because HIV is growing in immune cells, and it needs sort of semi-activated T cells to grow, but it doesn't want super aggressive T cells that are going to kill it. And so it was basically modulating T cells to function in a certain way. Okay. Now, when we started the research, and this, research was going from the mid-80s to the mid-90s, we thought what we were dealing with in HIV was autoimmunity, because here we have a piece of GP120 that looks human, and therefore maybe the immune system recognizes it as a cross-reactive epitope, and therefore this is something maybe we should suppress. Maybe there's a sort of disease process involved in HIV and cross-reactivity or cross-reactive antibodies. And this now jumps into a sort of general strategy we've always had, which is in times of extreme medical importance, and this time, early 90s for HIV was a time when there was very little in the way of therapeutic interventions available. Volunteers come to scientists and say, have you got anything in the lab? You know, maybe we could try it, maybe it would work. And I had that situation with an HIV patient in London. They came to us. At this point, I was running a joint venture between Medical Research Council, the University College London. In fact, there was a US element as well, Southwest Foundation for Biomedical Research in San Antonio, Texas. And anyway, we had this big collaboration team. A volunteer came to us. I said, look, I can't supply you with an experimental medicine directly, but I can give you the information on how to produce the peptide, where to get it taken. And also this volunteer then was actually attending clinics at Ealing Hospital in London. So we had there's a medical team who could basically see how things were going. And a very quick end to that story is he took the peptide, which is human Ezrin Peptide 1. It was observed a very rapid fall in his HIV viral load, a correction in CD4 and CD8s, and it looked like a therapeutic molecule. And so that was the start of a development program.
Yeah.
So fast forward 25 years into the future, and now, the present.
Long COVID.
So we're looking at long COVID. And,
yes.
And
so just so everybody knows how we're defining long COVID here. It's just the persistence of symptoms after somebody recovers from a COVID-19 infection. And
that is correct. So
right.
that, normally it's people who if you have an acute infection, it may persist for, say, 2 or 3 weeks. A lot of people who recover quickly might be clear in 1 week, but there are patients out there where it persists for 12 weeks, for 6 months, for 1 year, for 2 years. And generally, they call anyone who's still got symptoms after 12 weeks has got long COVID. Or another expression used in the States is sort of sequelae of COVID, PASC. And basically that is describing anything that's going on after an infection for the following 2 years. The issue has become more obvious over time. And now, for example, here in the UK, it's estimated there's about 2 million people who are manifesting long COVID symptoms. And not a full spectrum, not total severity, but there are a lot of people, and that's a very significant percentage of the population, who are developing or have maintained these sort of inflammatory symptoms.
Right.
Now, what happened was, beginning of this year, people began to realize this is a big prevalence. It's a major epidemiological question. What's going on with all these patients a long time after they had acute infection? And people started looking at it look at all the different things that happen to these patients. So generally, there's symptoms of inflammation, but it manifests in many, many different ways. So some people, it's just a continuation of their lung problems with difficulty in breathing, coughing, sore throat, those type of things. Others, there's systemic blood clotting or microclots occurring. There's inflammation in the myocardium, so people are starting to get cardiovascular problems. There's kidney problems. And then there's a large group of people who suffer from mental health issues where inflammation in the brain seems to be interfering with memory, with cognition. It also affects mood. So there's a very broad spectrum of effects. But what I looked at, I published a paper in Immuno, an MDPI publication in September, where I was trying to say, what is the sort of central core to all of these phenomena people are describing? And what I zeroed in on was, well, we already know from acute COVID that high levels of IL-6 expression are very bad for your general prognosis of outcome. Right. So if you're a patient who's been infected with SARS-CoV-2 and have a high level of IL-6, then you're more likely to have a severe encounter with the disease and maybe be hospitalized, maybe intubated, and whatever.
And just to stop you there, so some people may not know what IL-6 is. It's just, I guess we can call it a chemical
pro-inflammatory cytokine.
Right. A cytokine released when there's a lot of inflammation happening in the body. Okay.
Absolutely, great. Yes, so the issue was, okay, so we know in acute COVID, we get this high expression level of these inflammatory cytokines, pro-inflammatory cytokines. And not only IL-6 is the most visible one, but you also get IL-1B and TNF-alpha, and you also get IL-8 as well. All of those inflammatory cytokines play a role in this general upregulation of inflammatory processes. And the inflammatory processes, you've got to remember, are innate immune processes. These are very old processes. You go back to the beginning of vertebrates and before that, because they are pattern recognition. They're based on pattern recognition receptors. That is, organisms are pre-programmed to recognize invaders. So certain protein structures or carbohydrate structures are recognized by these receptors, and that kicks off a very powerful and rapid immune response. Now, we in our bodies have two systems. I'm going to get too sideways in this one. I'll try and keep it short. We have two basic systems. We have what is called innate immunity, which is a very old, evolutionarily conserved system, which has the toll-like receptors and quite well-known molecules in that direction. And then we have the adaptive immune system, which is something like where your body actually, in its lifetime, is programming appropriate responses, and that's based on T cells and B cells. Okay. Now, the problem with COVID is it's hyper-triggering that old innate system, and you get masses of inflammation. Now, depending on your genetic background, you'll get more or less of it. And unfortunately, the people who have super susceptibility have actually a larger component of Neanderthal DNA. This is one of the weird things that cropped up in research in the last 2 years. The basic idea, though, is that people have a genetic susceptibility to overactivation of their immune system, triggered by primarily spike protein on the surface of the SARS-CoV-2 virus. Okay, so that's the acute story. So you've got virus goes in, boots up this very powerful innate rapid response to an invasion, and overdoes it. It's overactive. Now, when the virus is cleared, that system should switch off, but unfortunately, it doesn't. And what it seems to happen is that it persists because there are certain feedback loops which get triggered by the infection. Okay, and when I was looking at, what are these persistent characteristics in these long COVID patients, yes, they're all of them showing a big spectrum of different types of inflammatory symptoms because of their genetic background. Some show one thing, some shows another. So somebody may have more of a sort of a cognition problem or a brain fog problem. Some might have more of a cardiovascular problem or even a gut inflammation problem. Okay, but the fundamental mechanism seems to be inflammation. And so I was able to propose that there's this family of proteins which is called the S100 proteins, particularly a protein called S100A8A9. Sorry to get too much into detail. But basically, when you get high levels of IL-1 and IL-6 signaling, chronic levels, you start to produce lots of this protein. It's actually being triggered by IL-6, triggering its own receptor on another cell, which then goes on to express this protein. The problem is this protein on its own will trigger IL-6 expression by binding to these two other innate immune receptors called TLR4, that's toll-like receptor 4, and RAGE, which is a great name for it because it's what's happening. Your system is raging. But it comes from actually a receptor for advanced glycation end products. It's a receptor generally associated with inflammation in aging, okay? But what I'm saying, this has the potential to create a closed loop so that the system is asking for more inflammatory cytokines. Those inflammatory cytokines produce more S100 proteins. S100 proteins hit the TLR4 receptors, and it goes round and round and round. And so,
okay, so just to summarize, in plain language, we have basically this inflammatory loop that just keeps going around and around, and it's not turned off. That's the theory for why the long COVID symptoms exist.
Exactly. And the thing is, that system is ubiquitous across the whole body. So it affects all organ systems.
Okay.
And the point is that even though the pro-inflammatory cytokines are much, much more active, I mean, they work at the sort of picogram per milliliter level, this stuff works at sort of micrograms per milligram per milliliter level. So what I'm saying is you've got to have a lot of it before it really pushes your body to be constantly inflamed.
Okay. And in
But
some of the studies in your paper published in Immuno in 2022, recent paper, there were some studies done where they measured the cytokine levels in people who had long COVID, right? They did actually do some. Okay, so just trying to get to like some of the evidence that supports this theory.
There's a number of stuff. I mean, certainly it's very clear already that IL-6 and IL-1 are very, very important in this process. It was less clear, the S100 proteins, but there's more and more evidence coming out that S100 proteins are involved in the long COVID persistence. I mean, it's already, these proteins, they have other names. There's some called calprotectin. They're actually calcium binding proteins. But they are definitely shown to be associated with outcomes with acute COVID. So there's lots of papers now showing that you can measure high levels of these S100, A889 proteins. It's not good news for your, prognosis, whether you're going to survive. But the issue I'm saying is that this is also important for long COVID. Okay, and so there are a lot of people still looking for basically persistent virus to explain long COVID, saying some reservoir somewhere in the body, it's low-level replications triggering that inflammation. But you don't need any virus. If the spike protein has triggered the system so much to actually produce lots of this S100 protein, you don't need anything else.
And that's also, and correct me if I'm wrong, in your paper you wrote that in at least half of patients who had long COVID, they didn't detect the genetic material, the genetic material from the SARS-CoV-2. Okay, so
that's right.
Okay.
Well, and the thing is, even when they do detect material, PCR data, either data from PCR or from looking at protein fragments or epitopes, you don't know whether you're looking at a functional protein or not. So it doesn't necessarily mean you're looking at a virus. It could be what's left of the virus.
I see. Right. Sure. Okay.
So what I'm saying is, as soon as you've got a disease where only half the patients show any evidence they've got virus there, then you should be looking for another mechanism. You should be trying to find what other fundamental reason why something's not working or why this thing is persistent. Okay, and there have been a number of experiments where people have tested various inhibitors, like Toll-like-4 inhibitors or RAGE inhibitors, in animal models. So they're looking in mice where they've got humanized ACE, so basically they get infected with the human virus, ACE being the primary receptor for SARS-CoV-2. And they've shown that indeed, if you inhibit either TLR4 or you inhibit RAGE, you will get some reduction in the inflammatory effects that you see, in these models. Okay? Now, I can jump to Ezrin peptides. Why are Ezrin peptides important?
Yeah, let's do that. So you've been studying these for a long time, and let's kind of summarize what you said at the beginning, or for people who may have joined in late or whatever. You wrote in your paper that Ezrin peptides may be a potential therapy for long COVID. Obviously, there's a lot of interest in how we're going to treat long COVID. It's a big area of research right now. You've been studying these a long time. I know you explained in the beginning of the podcast, but can you just give us the elevator pitch,
All right,
as simple as you can for people who may not, be in science or whatever? Like, what is this Ezrin peptide?
All right. Ezrin peptides are synthetic copies of a bit of a protein called human Ezrin. Human Ezrin is an essential component of cell signaling mechanisms from receptors down into the cascades that phosphorylate all these other proteins. Okay? We started making synthetic Ezrin peptides because we thought we were trying to induce immunological tolerance when we were working in HIV. But during our clinical development work, and in fact, this is where I sort of jumped out previously, I was first doing the work in London in the US, and then I started doing it in Moscow. And the reason that happened was in the mid-90s, suddenly everyone was interested in Russian science. And so I set up operations inside this immunological institute called the Gamma Layer Institute, which later became famous because of the Sputnik 5 Russian vaccine, the adenovirus vaccine. Okay? And in fact, some of those people who were working on that vaccine were also working with my team on some things we were doing in these other areas. In fact, we actually built a pharmaceutical company.
And the Sputnik 5, that's the Russia's vaccine for COVID-19.
That's the vaccine for COVID-19. It's an adenovirus vaccine. So it's adenovirus expressing a bit of actually spike protein.
Okay.
Receptor binding domain of spike protein.
Right. Okay.
Anyway, so I didn't ever expect, my Russian activity to become a dominant activity, but that's what happens. And in fact, Russian immunologists are really excellent at doing workup and exploring opportunities and possibilities. And so when we were doing development with Ezrin Peptides, which was originally developed for HIV, we discovered, or my collaborator, Professor Ravshan Atalakhanov of the Institute of Immunology, discovered that in fact, these Ezrin peptides have a very strange property. That is, they will inhibit the expression of inflammatory cytokines, but at the same time will amplify B and T cell immunity. Okay, so what it's doing is suppressing innate immune responses and upregulating adaptive immune responses. Very simple.
Okay, and innate is just the non-specific immune response.
Innate is a genetically pre-programmed response, which is inflexible. It either is on or off, right? So if beta is recognized, bang, you get high reactivity, whereas the adaptive response is all your T and B cells and your B cell receptor and T cell receptor. A little more actually recognizing something in your own lifetime.
You're tailored to the enemy invader, so to speak.
Yeah, exactly. So it's like, much more better sort of signals intelligence.
Right.
And yeah, so that's what we had. And then in the clinical trials, we first started working on HIV, and we saw our best results were in fact in HIV opportunistic infection. Okay. And in fact, we got a phase 2 approval just simply based on that data, because we'd already demonstrated these peptides are really safe. You just don't get any toxicity. You sort of expect that peptides, in terms of their drug metabolism, because they get broken down very easily, but still a highly active peptide can still do a lot of damage if it's triggering something you don't want it to trigger. But it turns out these Ezrin peptides are highly regulated, so they have unusual bell-shaped dose-response curves. That means you're operating inside a regulated system, whereas often when you have a small molecule drug, you keep putting it in, something happens more and more and more until you get toxicity. Okay, so what we're saying is that the Ezrin peptides are part of a regulated immune network of activity. And okay, this is not just scientific speculation. We started to do more clinical studies, because as soon as we could see we could knock out opportunistic infection. Now, the one thing that AIDS patients or HIV patients get is severe candidosis, candida infection, fungal infection. So we started working in chronic candida with female patients who've got STD candida or sort of chronically immune suppressed and therefore getting candida.
Yeast infections, right?
Yeast infections, yes.
Okay.
And so we found we could very rapidly stop those infections. I mean, literally within like 3 doses over 5 days, and intravaginal doses. And what we observed, or the clinicians observed during those studies, was that there were definitely that peptides, these Ezrin peptides, reduce the inflammation of the mucosa at the same time as increasing an immune response to fight the infection. And the best thing was they were also inducing a long-term immune protection. And so what we found with candida is that Ezrin peptides act a bit like an adjuvant. So once you've had the infection, you don't get that one again. Okay, if you change partners and you've got a different strain, then you might get it again. But at the moment, cancer is a huge problem still, because antifungals control it but don't eradicate it. I mean, in US, for example, cancer is a huge thing. It's endemic, big problem. Anyway, so moving on from cancer, cancer is the first example. We started, again, this is what's beautiful about the sort of Moscow clinical network. When people hear something's working and it's safe, then everyone starts trying it and seeing if it works in their particular medical problem. And so we had people testing it for herpes, for human papillomavirus, for rotavirus, which is a gut virus causing diarrhea, big spectrum of viral infections, and also to a lesser extent, bacterial infections, and basically getting the same type of results. That is, you are reducing inflammation, but improving the adaptive immune response. We even did a human clinical study with hepatitis B vaccine, where when you take, this is HEP1, the first version of this peptide, you could increase the titers of the hepatitis B antibodies, by combining the vaccine plus peptides. Okay? So the general property seems to be immune amplification of the adaptive system, but suppression of the pro-inflammatory system, which is the innate system. That's very generally what's happening. And so there's some switching process, which we're still trying to fully understand. But what it seems to be is that generally the inflammation is being mediated by a transcription factor called NF-kappa beta. And so most of the inflammatory cytokines get expressed when that transcription factor gets upregulated. However, there's other transcription factors, and some of those will compete. So some of it, like this IL-10, which is actually a suppressor of inflammation, and that will do the reverse, and it uses a different transcription factor. So one of the things we're investigating is Ezrin, changing the conformation of Ezrin complexes, protein complexes, changing transcription factor usage, and that might explain why you're getting this. It's like a complete shift in the way immune cells work. So they go from one state to another state. Okay, so we have this sort of technology, and again, the story expands because I'm talking here about 25 years of clinical research. We started to notice when you're giving these peptides orally to patients for whatever reason, if they had oral ulcers, mouth ulcers, they would disappear. And we thought, wow, that's interesting. Why is that happening? Well, we sort of understood if you get the inflammation down, that's good. But we also started to get data on tissue regeneration. That is, these peptides act on fibroblasts and stimulate some types of tissue repair action as well. So I'm beginning to make this thing look like elixir of life, but it has this multiple activity because it's part of our system. Right? It's a central control point of our system. Okay? You could say it's general, and very generally, sort of anabolic growth, defense, those type of functions. Okay? So when we did experiments with the gut ulceration, we started to work with duodenal and stomach ulcer patients, and we started getting similar effects with quite rapid reduction in ulcer problems. And also, again, protection. This is the other thing that's very interesting. Is people get stomach ulcers often get recurrent stomach ulcers. It's not clear why. It may be due to H. pylori, which is a bacteria which is associated with these gut ulcers. It may be some other sort of stress factors that affect with the mucosa. Anyway, it's like a black box. We don't know, but we do know that if you take these peptides, you have some sort of long-term protection against these ulcerative problems. And we did do one study in IBD. So we had lower gut ulcer.
Irritable bowel syndrome?
Irritable bowel syndrome.
Okay, another common.
That's important. So I'm going to jump back to long COVID now, because some groups of long COVID patients also get these gut problems. They get these inflammatory gut problems. And in fact, I've just had a volunteer who's come to me who said he basically had long COVID. He got an immune booster from one of the vaccines, and I don't know which one he used, and then got more severe inflammatory bowel problems. We're really interested to see, because he's going to try the newer version of the peptide, just see if we get any effect at all. And with all these volunteers, we always start at super low dose. At the beginning, if something goes well, that's great, but we're always doing that to make sure we don't make things worse for a patient. But at the moment, we do have evidence from certainly one ME long COVID patient, a combination patient, who seems to be responding well to the RepG3 peptide. This is a later peptide. And we also have some data we have this German clinic who's been working with these peptides, and they have some claims of cures. I'm not saying big numbers, it's probably about 5 patients. I'm not sure exactly how numbers, but where people have presented long COVID, they actually received Ezrin peptide as an injection in low dose and had a resolution.
So now, you can take them orally or that's what you're studying? Okay.
Yeah, my team's generally working with oral or spray inhalation. And then we've tended to go in that direction on spray inhalation because you don't need that much. Literally you need about 0.5 of a milligram for a 70 kg person to get an effect. It's quite active. And so, in fact, if you spray and inhale, then you just coat the mucous membranes. It's not going down to the stomach, and it's just going in at that point. And we think a lot of the effects are secondary signaling. So you're getting an effect to the membrane, and then you get secondary cytokine interferon effects and a number of other effects.
Okay.
So that's the general idea.
The general. So like in a quick sentence for how you were pitching this for long COVID, it sounds like the fact that long COVID affects multiple organ systems and these can act in multiple organ systems, they shut down. Yeah. Go.
Well, the critical thing is, okay, first, long COVID is a disease at the moment, or a syndrome where people might have to take medication for a long time.
Yeah.
You can't immunosuppress people for a long time because then they've got other infections. And if it is, that's what I'm saying. The core disease mechanism is sort of self-perpetuated immune stimulation. I mean, pro-inflammatory stimulation. You've got to shut that down, but not shut down the whole of the immune system, because then and in fact, this was, I don't know if you heard the story about the black fungus in India, because there were
I
people,
did. Yeah.
And that was due to people taking corticosteroids, thinking, okay, that will reduce the risk of acute COVID and the inflammation there. But of course, then you expose yourself to all sorts of nasty infections around there. And so that's what I'm saying. It doesn't have to be our stuff, which is the solution, but the solution, the characteristic of the solution must be to shut down whatever that inflammatory, innate, aberrant inflammation is without knocking out the whole system. And that's the big task, medical task, to work out how to do that. We are lucky because we've got a molecule or a group of molecules where we already knew they do that. Right? And in fact, the reason, we started treating acute COVID, I must go back. I'm not vaccinated, okay? I'm not an anti-vax person. I'm just not vaccinated because I cured my own acute COVID in March 2020, and I've never got it again. I was jumping back between Moscow and London and Cyprus and everywhere else. And I was constantly being PCR tested, did not have SARS again, or, there was no detection of PCR. And so again, I'm saying what these proteins, these peptides are doing is suppressing these inflammatory processes that make you sick, but not suppressing your immune system. In fact, they are amplifying your immune responses.
Okay. And so you mentioned some volunteers and, who came forward to try this for their long COVID symptoms, some preclinical data. Obviously,
we have to be very careful how we tread, because the reality is, if you're going to run a clinical trial, you must go through the whole regulatory process.
Yeah.
With Ezrin Peptides, they went through with Ezrin Peptide 1, it went through the whole regulatory process in Russia. So it's freely available in Russia for treatment of COVID and long COVID and everything else. Yeah? Anywhere else, if they have legislation that allows a doctor to make a prescription based on a registration in the country, then that's also okay. But even, for example, in Europe, unless you've done a full preclinical toxicology, a big package of data, you're not supposed to do any clinical trials. The only way you can work with patients if they are individual volunteers and they have an unmet medical need. And in this case, this is what's happening in long COVID. And it happened, of course, in acute COVID. And even now with, okay, yes, there are a bunch of antivirals out there that may work, but the evidence is building up. The antivirals aren't that effective for acute COVID. And there are problems associated with vaccines. So we still need to find solutions.
Right. But from what I've read from your paper, it seems like you want to do a larger experiment just because people want, I think, the benefit of that is just, I know that we have this red tape and regulation, and sometimes red tape can be really frustrating, but it's there for
it's essential to protect the population.
Yeah.
Crazy people,
well, sure.
dangerous medicines. Yeah.
Sure. But there's a reason, there's a method here. You want to make sure it works, and you want to test it against a placebo. And I didn't know if that's what you were planning. It sounds like you're at a company that makes these.
Well, we're very small scale at the moment in the UK.
Small scale. Okay.
Yeah. Because I was managing, 1,000-person company in Moscow until January. Okay.
Oh, okay. So until
And
fairly
then, of
recently.
course, you know, boom, world changed. I'm now in London, and NewalR&D, my research company, is a very small company, and we've just got our seed funding. We're looking to raise. We need maybe up to 10 million UK pounds to do a full clinical development program, because we have, you know, we've got all the data on the earlier molecule. The new
Right.
molecule is actually just small modification on the older molecule, just upregulates its anti-inflammatory properties. But yeah, we need to be properly funded and do large-scale studies. I mean, just normal formal registration studies, and that's where we're going.
That's where you're going.
Yeah.
Okay.
But
okay.
the thing is, the advantage of the way we do things is that we know in advance that, okay, we've got a few patients out there who seem to respond well to this therapy, and it's also, we've got actually a huge database of safety data in the sense we know this class of compounds don't do nasty things to you, and over a long period of time. So it's like, you know, we've got at least 50,000 people over 25 years, which is pretty good. You know, so you find, you know, if you're going to have a problem, we'd have seen it by now. So.
And so the next steps is doing those studies. And I just want to make it, you talk about the different types of Ezrin peptides, or different generations. I know you had the HEP, was that the first one? The human
HEP1. Human Ezrin Peptide 1, you know,
and then there's
boring scientific names. That's how they do it. You know.
As long as the names are easy to remember.
We've got basically four generations, because the sequence is that, okay, we found this bit of human Ezrin which has been copied by HIV. So the first molecule was a 14 amino acid peptide, and the reason we chose that size is it's a MHC class 2 antigen presenting size. This is the size of peptides that are normally presented in the adaptive immune system. Okay. When we started working on the basic science of Ezrin peptides, we were trying to work out what the receptor was, because once we understood we weren't dealing with autoimmunity, we were trying to work out what the receptor is. And as far as we can see, the receptor is Ezrin itself, right? So Ezrin is actually it changes shape in response to these peptides. And so what we actually did was then start to break up bits of the alpha domain of Ezrin and look at other sequences, because basically HEP1 is a very charged molecule. It's sort of plus minus, plus minus, plus minus. It will very much interact with another protein. It forms an alpha helix. Okay? In fact, on my coffee mug, that's it. That's the thing. That's what it looks like.
Great mug.
It's a coffee mug. No, it's a sequence. And that's an alpha helix. It's like 4 turns, 5 turns from alpha helix. Right. And that's what it is. Yeah, so what we did on the generation 2 was we looked at bits of that region of Ezrin, so chopping them up, synthesizing bits and putting them into the same assays. And we actually found that a lot of peptides from that region of Ezrin are biologically active in a similar way. Okay. We then start, and these are but still natural sequences. Okay, generation 3, which is this thing we call RepG3, which is one that people are using in long COVID right now, is a small modification of HEP1, where we chopped off some of the charged side chains of HEP1, because we were sort of thinking, basically, if you have a super high charge on a molecule, it doesn't get into cells very easily. So what we thought, if we could just remove, down the polarity of the terminal bits of the peptide, maybe to help the activity. And indeed it did. And we seem to upregulate the anti-inflammatory effect. It didn't seem to change the adaptive immune amplification effect. But this is just simple experimentation, playing around.
Right.
The generation 4 are just other, synthetic varieties of these molecules we already know about.
And just to clarify, these are approved, or some of them are approved for use in Russia?
HEP1 is approved in Russia. So first molecule is approved in Russia. Everything else, they're experimental molecules, but they are very closely related to the approved molecule.
Okay, and Russia's the only country right now where,
at the moment, unfortunately, yeah.
Okay, official approval. Okay. And
because it's basically a problem of funding. I mean, okay, if you're a Pfizer, you can go worldwide very quickly.
Well, money,
yeah, but
you need money to run experiments.
If you're a small research team,
of course,
it's one country.
No, of course, if something doesn't get the funding, it doesn't get the required studies that it needs to get that approval, right? If it shows a positive result, it's Funding is so important.
Yeah, and the basic economics actually of drug development are, okay, when you'll see analysts' reports of it now costs $1 billion to develop a drug. That's not true. I mean, that covers all the costs of the infrastructure, the failures, the huge sort of like mess of investment that goes into drug development. The direct cost of getting a drug out is about, well, we calculate about £30 million. It depends on one of the big costs is if you've got to build a factory before you can test the drug. That actually is true now for EU regulations. You must have GMP production before you can go into patients, for example. But that's that's generally the costs that you need. Say approximately 30 million. Okay, maybe it's 50, maybe it's 20, but it's that ballpark. It's not $1 billion basically to get a drug out.
Interesting. So that was really fascinating stuff. And it sounds like you're probably going to explore using these peptides or continue to explore using these peptides for more than long COVID.
Yeah.
I wanted to focus on long COVID for this podcast because that's obviously a hot topic, and a lot of people are out there suffering with long COVID symptoms. So it's interesting to hear about different things that may work. If people are interested learning more, maybe getting in touch with you, talking about partnerships, whatnot, getting information on Ezrin peptides, where's the best way how can they do that?
Okay. Well, on my publications, I haven't put my phone number there and my email's there. So anybody who wants to sponsor work, great, just give me a call.
Give you a call. Okay.
And I actually have a website, a personal website, which again has a contact point. In fact, I get a lot of my volunteers from that personal website. They say, like, we've got no solution, we'd like to try this. And so, but the volunteer program is tiny. Like with me, it's sort of like, was 3-4 out there right now. The German group has a larger group of people they're treating, but it's probably in the tens. I don't think it's more than 100. They're treating on the basis of this registration in Russia, and it's allowable in Germany for single patient use. But the bottom line is we need properly funded studies, otherwise it's never going to reach a large number of people.
Right. Yeah. So hopefully, I think it's a really fascinating area. I definitely learned a lot, and I want to wish you a lot of luck, and hope if you any more research or interesting findings, feel free to come back on the Trial Site News podcast. And
brilliant. Okay. Yeah. Well, I'll let you know. I'll keep you in touch.
Absolutely.
Things evolve.
Yeah. No, of course. And there's a lot of people who are interested, especially in ways to address long COVID. So really interesting.
But, maybe I can close on one big concept, which is that long COVID is being described by clinicians with its multiplicity of symptoms. Okay, but a lot of it is associated with age-related disease. Okay, so you're seeing arteriosclerosis, arthritis, inflammatory bowel syndrome, lots of things. Now, if it may turn out that a long COVID solution will have solutions in other therapeutic areas automatically, because we'll see those comorbidities going down.
Sure.
Right? So that's a big opportunity and hope for what could go on in this area.
Sure. No, that makes sense based on what you've described. And I will include links to your website and publications and anything else that you want in the podcast description. So for our viewers, you could see that in the description there on YouTube. And yes, thank you.
That would be wonderful.
Absolutely.
But if I get deluged with volunteers, I really can't manage it. I can do a few. I can't do hundreds. Definitely not.
Oh, if people reach out to and they want to be volunteers. Okay. Well, you might get some phone calls out. You did put that out there. So
okay, it's true. But just a modest number, not sort of like crowds in front of you.
Your phone might be ringing off the hook, so to speak. Okay.
All right, Erin, thank you very, very much.
Thank you so much, Dr. Holmes, for your time, and enjoy the rest of your day. And thank you to everybody for joining in today on Trial Site.
Okay, thank you. Goodbye.
Absolutely.